We will now do Week 5, Patterns of Single Gene Inheritance, Part 1. The learning objectives for this week are to discuss the role of genes in the production of proteins, explain the mechanism of Mendelian inheritance patterns, autosomal dominant, autosomal recessive, X-linked recessive and dominant, and Y-linked, and to discuss genetic heterogeneity, penetrance, expressivity, anticipation, chimerism, imprinting, mosaicism, and consanguinity. The genetic information is contained in the DNA in the chromosomes within the cell nucleus. Protein synthesis, the process through which the information encoded in the genome is actually used to specify cellular functions, takes place in the cytoplasm. The DNA of the human genome, which encodes the protein, is called the exome. However, this represents only a very small part of the genome. Much of the human genome controls protein synthesis, including the time, speed, and locations. The genes encode approximately 20,000, a little over 20,000 types of proteins. If you look at this slide, you can see some of the protein diversity in the human body. You can see the different types of proteins and what their function is. As we will be going through this course, we will be learning about individuals who do not have specific genes and the effects that this can cause, because one protein can cause a disease. If we look at the production of protein from the instructions that are contained on the DNA, this gene expression requires several steps. Transcription, the synthesis of an RNA molecule. Translation, where you use the information in the RNA to manufacture a protein by aligning and joining specified amino acids. And then folding of the protein into a specific three-dimensional form. Shortly after Watson and Crick published their structure of DNA in 1953, they described the relationship between nucleic acids and proteins as a directional flow of information called the central dogma. As Francis Crick explained in 1957, the specificity of a piece of nucleic acid is expressed solely by the sequence of its bases, and this sequence is a code for the amino acid sequence of a particular protein. This statement inspired more than a decade of intense research to discover exactly how cells make proteins. The process centers around the RNA. If we look at RNA structure and types, RNA is the bridge between the gene and the protein. The bases of an RNA sequence are complementary to those of one strand of the double helix, and this strand is called the template strand. RNA polymerase builds an RNA molecule. It's the non-template strand of the DNA double helix is called the coding strand. There are two types of nucleic acids, RNA, DNA. Both consist of sequences of nitrogen-containing bases joined by a sugar phosphate backbone. However, they do differ in several aspects. This is a slide showing how DNA and RNA differ. In DNA, it's usually double-stranded. RNA is usually single-stranded. Thymine is the nitrogenous base in DNA, and uracil is the base in the RNA that replaces a thymine. Deoxyribose is the sugar in DNA, while ribose is the sugar in RNA. The DNA maintains protein encoding information, but the RNA carries protein encoding information and controls how the information is used. DNA cannot function as an enzyme, however, RNA can function as an enzyme. DNA persists, whereas RNA is only transient. Here we have showing you the structures of the DNA. You see the double-stranded versus the single-stranded. You see the deoxyribose is the sugar in the DNA. The ribose is the sugar in the RNA. Here you see that the bases that are found in the DNA are the thymine, cytosine, adenine, and guanine, and the bases in the RNA, uracil, cytosine, adenine, and guanine. There are three major types of RNA: messenger RNA or mRNA, ribosomal RNA or rRNA, transfer RNA or tRNA, and other classes of RNA control gene expression. Here we have a slide showing you the size and function of the messenger RNA, the ribosomal RNA, and the transfer RNA. Let's look at messenger RNA. It carries the information that specifies a particular protein. 3 messenger RNA bases in a row form a codon, which specifies a particular amino acid. There are 20 amino acids. Most messenger RNAs are 500 to 4,500 bases long. Differentiated cells produce certain messenger RNA molecules called transcripts, and the information in the transcripts is used to manufacture the encoded proteins. This slide shows the relationship among RNA, the DNA template strand, and the DNA coding strand. The RNA sequence is complementary to the DNA template strand and is the same sequence as the DNA coding strand, with uracil, U, in place of thymine, T. rRNA, ribosomal RNA. Most RNAs are about 100 to 3,000 nucleotides long. They associate with proteins to form ribosomes. The ribosomes consist of 2 subunits that join during protein synthesis. rRNAs provide structural support. Some actually work as catalysts, they're ribozymes, and others help align the ribosome and messenger RNA. Looking here at the structure of the ribosomal RNA, it's the ribosome from a eukaryotic cell, and it has 2 subunits together. They consist of about 79 proteins and 4 rRNA molecules. tRNA binds a messenger RNA codon and a specific amino acid. It's only about 75 to 80 nucleotides long. The 2-dimensional shape is a cloverleaf shape. The 3-dimensional shape is an inverted L. It has 2 ends. The anticodon is complementary to the messenger RNA codon. The opposite end strongly bonds to a specific amino acid. If we look at this slide, the very top, we see the A. And what A is representing is that certain nucleotide bases within a transfer RNA hydrogen bond with each other, giving the molecule a cloverleaf conformation that can be represented in 2 dimensions. The darker bases at the top form the anticodon, the sequence that binds a complementary messenger RNA codon. Each transfer RNA terminates with the sequence CCA, where a particular amino acid covalently bonds. In B, this represents a 3-dimensional representation of a transfer RNA, and it depicts the loops that interact with the ribosome. On the bottom, you can see a transfer RNA with a particular anticodon sequence and how it always binds the same type of amino acid. If we look at transcription factors, we find that they interact and form an apparatus that binds DNA at certain sequences, initiates a transcription at specific sites on chromosomes, respond to signals from outside the cell, and link the genome to the environment. Mutations in transcription factors may cause a wide range of effects. Looking at the steps of transcription, transcription is described in 3 steps: initiation, elongation, termination, and in initiation, transcription factors and RNA polymerase are attracted to a promoter. The RNA polymerase joins the complex, binding in front at the start of the gene sequence. In this slide, you can see in A, the proteins that initiate transcription recognize specific sequences in the promoter region of a gene. In B, a binding protein recognizes the TATA region and binds to the DNA. This allows other transcription factors to bind. And in C, the bound transcription factors form a pocket that allows RNA polymerase to bind and begin making RNA. Looking at the steps of transcription, in transcription elongation, enzymes unwind the DNA double helix. Free RNA nucleotides bond with exposed complementary bases on the DNA template strand. RNA polymerase adds the RNA nucleotides in the sequence the DNA specifies. A terminator sequence in the DNA indicates where the gene's RNA encoding region ends. Here we have a picture showing the transcription of RNA from DNA. The transcription occurs in 3 stages, as we discussed: the initiation, elongation, and termination. Initiation is the control point that determines which genes are transcribed. DNA nucleotides are added during elongation, and a terminator sequence signals the end of transcription. This slide shows that many identical copies of RNA are transcribed simultaneously. Usually 100 or more DNA bases lie between RNA polymerases. In eukaryotes, such as humans, messenger RNA must exit the nucleus to enter the cytoplasm. Several steps process pre-messenger RNA into mature messenger RNA. A methylated cap is added to the 5' end. It is a recognition site for protein synthesis. A poly-A tail is added to the 3' end. And this is necessary for protein synthesis to begin, and it stabilizes the messenger RNA. When we look at the next part, the splicing occurs. The introns are the intervening sequences. They're removed, and the ends of the remaining molecules are spliced together. Exons are parts of messenger RNA that remain and are translated into amino acid sequences. Note that introns may outnumber and outsize exons. Messenger RNA is proofread, and the mature messenger RNA is sent out of the nucleus. If you look at alternate splicing, it's the mechanism of combining exons of a gene in different ways. Cell types can use versions of the same protein in slightly different ways in different tissues. This slide is showing you messenger RNA processing, the maturing of the message. In this slide, several steps process pre-messenger RNA into mature messenger RNA. First, a large region of DNA containing the gene is transcribed. Then a modified nucleotide cap and poly-A tail are added, and introns are spliced out. Finally, the intact mature messenger RNA is sent out of the nucleus. During translation, you assemble a protein using the information in the messenger RNA sequence, and particular messenger RNA codons correspond to particular amino acids. This occurs on the ribosome. And in this picture here, for translation, you can see in this process that the messenger RNA is transcribed from a locally unwound portion of DNA, and in translation, transfer RNA matches messenger RNA codons with amino acids. The genetic code is the correspondence between the chemical languages of messenger RNA and proteins. In the 1960s, researchers used logic and clever experiments on simple genetic systems to decipher the genetic code. The researchers established certain requirements for the code. Here we have the genetic code in this diagram right here, just showing you how it was put together. The genetic code is a triplet code. 3 successive messenger RNA bases form a codon. There are 64 codons. Altering the DNA sequence by 1 or 2 bases produces a different amino acid sequence due to disruption in the reading frame. Adding a base at one point and deleting a base at another point disrupts the reading frame between the sites. Adding or deleting 1 or 2 nucleotides in a DNA sequence results in a frame shift that disrupts the encoded amino acid sequence. Adding or deleting 3 bases does not disrupt the reading frame because the code is triplet. This is a simplified representation of the Crick experiment. The genetic code is non-overlapping. In an overlapping DNA sequence, certain amino acids would follow others, constraining protein structure. The genetic code includes controls. It includes directions for starting and stopping translation. An open reading frame does not include a stop codon. Even though the genetic code is non-overlapping, any DNA or RNA sequence can be read in 3 different reading frames, depending upon the start base. If you take a look at this slide, you can see that it depicts the three reading frames for the sequence AUG, CCC, AAG, with AC making it more extended. It encodes three different trios of amino acids. The code is also universal. There's evidence that all life evolved from a common ancestor. Different codons that specify the same amino acids are termed synonymous codons, and non-synonymous codons encode different amino acids. Therefore, in translation, where you're building a protein, it requires messenger RNA, tRNAs with amino acids, ribosomes, energy molecules such as ATP and GTP, and protein factors. Is divided into three steps: initiation, elongation, and termination. The translation initiation is where the leader sequence of the messenger RNA forms hydrogen bonds with the small ribosomal subunit. The start codon, AUG, attracts an initiator transfer RNA that carries methionine. This completes the initiation complex. Here, when you look at this slide, you can see where the translation begins as the initiation complex forms. And the initiation of translation brings together a small ribosomal subunit, the messenger RNA, and initiator tRNA, and aligns them in the proper orientation to begin the translation. In the translation elongation, the large ribosomal subunit joins. The GGA binds to its complementary anticodon, which is part of a free transfer RNA that carries the amino acid glycine. Two amino acids attached to their transfer RNAs align. And in the process, the positions of the sites on the ribosome remain the same. They cover different parts of the messenger RNA as the ribosome moves. The P site bears a growing amino acid chain. The A site holds the next amino acid to be added to the chain. Amino acids link by a peptide bond with the help of ribosomal RNA that functions as a ribozyme. The polypeptide builds one amino acid at a time. Each piece is brought in by a transfer RNA whose anticodon corresponds to the consecutive messenger RNA codon as a ribosome moves down the messenger RNA. When you're looking at this picture of the slide building a polypeptide, you can see that in a large ribosomal subunit binds to the initiation complex, and a transfer RNA bearing a second amino acid, in this case glycine, forms hydrogen bonds between its anticodon and the messenger RNA's second codon at the A site. The first amino acid, methionine, occupies the P site. In B, the methionine brought in by the first transfer RNA forms a peptide bond with the amino acid brought in by the second transfer RNA, and a third transfer RNA arrives, in this example carrying the amino acid cysteine, at the temporarily vacant A site. In C, a fourth and then fifth amino acid link to the growing polypeptide chain. The process continues until reaching a stop codon. For termination, it occurs when the stop codon enters the A site of the ribosome. A protein release factor then frees the polypeptide. The ribosomal subunits separate and are recycled. A new polypeptide is released. In this picture, you can see what happens during the terminating translation. And in a protein release factor binds to the stop codon, releasing the completed polypeptide from the tRNA. And in B, you can see freeing all of the components of the translation complex. This portion here shows that several ribosomes can simultaneously translate a protein from a single messenger RNA. These ribosomes hold different sized polypeptides. The closer to the end of the gene, the longer the polypeptide. Proteins called chaperones help fold the polypeptide. In looking at the protein structure, the proteins fold into one or more three-dimensional shapes or conformations. Based on attraction and repulsion between atoms of proteins and interactions of proteins with chemicals in the environment, there are 4 different levels for protein structure: a primary structure, a secondary structure, a tertiary structure, and a quaternary structure. In looking at the folding of the proteins begin to fold after the amino acid chain winds away from the ribosome. The first few amino acids in a protein are secreted in a membrane and they form a signal sequence. This leads it and the ribosome into a pore in the endoplasmic reticulum membrane. It's not found on proteins synthesized on free ribosomes in the cytoplasm. The chaperone proteins stabilize the partially folded regions in their correct form. They prevent a protein from getting stuck in an intermediate form, and they're developed into drugs to treat diseases that result from misfolded proteins. Misfolded proteins are tagged with ubiquitin, and the protein with more than one tag is taken to a proteasome, a tunnel-like multi-protein structure. As the protein moves through the tunnel, it is straightened and dismantled. Proteasomes also destroy properly folded proteins that are in excess or no longer. In this picture, what you can see is that ubiquitin binds to the misfolded protein and escorts it to the proteasome. The proteasome, which is composed of several proteins, encases the misfolded protein, straightening it and dismantling it. Proteins misfold in 2 ways: from a mutation, where a mutation alters the attractions and repulsions between parts of the protein, or by having more than one conformation. A prion protein can fold into any of several conformations and be passed on to other proteins upon contact, propagating like an infectious agent. In several disorders that affect the brain, the misfolded proteins aggregate, and they form these masses of proteins. And these protein masses inhibit proteasomes so that they cannot process any malformed proteins. Different proteins are affected in different disorders. This is a chart showing disorders associated with protein misfolding. One important thing to discuss is, however, that it isn't always clear whether the accumulated proteins cause a disease or are a response to the disease. And we will be discussing this as we go further into the course. Just to take a look at one of the proteins here, if we look at Huntington disease, for example, extra glutamines in the protein huntingtin cause it to obstruct the proteasomes. I would like to end this part of this week's work by discussing just a little bit about prion diseases. They were first described in sheep, which develop a disease called scrapie when they eat prion-infected brains of other sheep. The name denotes the way the sick sheep rub against things to scratch themselves. Their brains became riddled with holes. More than 85 animal species develop similar diseases. The first prion disease was recognized in humans was Kuru, which affected the native Fore people who lived in the remote mountains of New Guinea. Kuru means to shake. The disease began with wobbly legs, then trembling and whole body shaking. Uncontrollable laughter led to the name laughing disease. Speech then slurred, thinking slowed, and a person became unable to walk or eat. Death came within a year. The disease, which affected mostly women and children, was traced to a ritual in which people ate their war heroes. When the women and children prepared the brains, prions entered cuts and they became infected. At first, the disease seemed to be inherited because it affected families. Not many people knew about this disease, but in the mid-1990s, prion diseases dominated the headlines when mad cow disease in the United Kingdom led to a human version called variant Creutzfeldt-Jakob disease. More than 120 people ate infectious prions in beef and became ill. The prions cause a disease both by spreading the alternate form, an infectious or mutant form of protein. These aggregates of protein are also seen in more familiar diseases such as beta amyloid plaques and tau protein neurofibrillar tangles in Alzheimer's disease. They're also seen in the alpha-synuclein deposits in the Parkinson's disease. This is a slide showing how prions change shape. In looking at the scrapie, the disease of the sheep, we see that a single scrapie prion protein contacts a normal protein and changes it into a scrapie conformation. As the change spreads, disease results. Accumulated scrapie prion proteins clog the brain tissue, eventually causing the symptoms.